Okay, so in this video here, we're going to do the DNA, manipulating the DNA assignment. This is web assignment 3. We are going to have to identify the tools of the trade. So as we go, I'm going to make a couple different stops to take note of certain items, such as the restriction enzymes, plasmids, ligase, and once we get to the techniques of the trade, transformation, gene cloning, PCR, electrophoresis, and DNA sequencing, we have to describe the purpose of each technique listed. Why is it used? So we first begin manipulating DNA. The manipulation of DNA is often the first step in many biotechnology applications, from research and development to biomanufacturing. In this lesson, we'll be introduced to the basic biological tools and techniques that allow scientists to study and manipulate a gene of interest. As you recall, genes encode proteins, so if we control genes and their expression, we can control the production of proteins, including proteins like insulin that can promote human health. So we move on here, some biological tools of the trade. So this is all part of the assignment that we've been given, actually. So our web assignment, we have to identify the tool itself and describe in general how the tool is used, and we have to do it for restriction enzymes, plasmids, and ligase. Okay, so DNA plasmid map, still image. A DNA plasmid map identifying sites where the DNA can be cut by restriction enzymes. So here, like for example, ECO0109141133, and this NARI, ECO-RI, it's got a different set of sequence of numbers. SAC-II looks like MLUI, BAM-HI, BG2, or I'm not sure exactly how they're referring to them, but these are the different sites. So these are the restriction enzymes where my mouse is, and where the line is, that's basically where they're going to cut. That's this is a DNA plasmid map, right? Identifying sites where the DNA can be cut by restriction enzymes. So we're going to just I'm going to take a note here, describe in general how the tool is used. So for restriction enzymes, I'm just going to put a little note here. Restriction enzyme, it cuts the DNA in a specific region of the plasmid cut the DNA in a specific region exclusive to the particular enzyme itself. Okay, so I'm just going to put that, we may reword it. Cutting and pasting DNA in 2D animation. So the discovery of enzymes that could cut and paste DNA made genetic engineering possible. Right, so it's cut the DNA in a specific region exclusive to the particular enzyme itself. Right. So restriction enzymes found naturally in bacteria can be used to cut DNA fragments. Okay, so we're going to take some info from this. So restriction enzymes found naturally in bacteria can be used to cut DNA fragments at specific sequences. I just took note of that. While on the other hand, another enzyme, DNA ligase, can attach or rejoin DNA fragments with complementary ends. So I'm going to take a little note of that, ligase. what's it do? Can attach or rejoin DNA fragments with complementary ends, right? So I'm also going to put a little note. The restriction enzyme, I'm just putting a note, temporary note here. Found naturally in bacteria. Okay, so restricted enzymes are sequence specific. They recognize and bind to specific DNA sequences. Okay, so that's another good note. They recognize and bind to specific DNA sequences. Okay, so we look at this. We have EcoRI, it binds right there. Once they bind to their recognition sequence, restriction enzymes cut the sugar phosphate backbones of the DNA strands. EcoRI is a restriction enzyme that cuts unevenly, leaving overhangs or sticky ends. Okay, so we have the sticky ends here. The sticky ends can be reattached by another enzyme called ligase. It catalyzes the chemical reaction that rejoins the DNA sugar phosphate bonds. Sticky ends can be reattached by another enzyme called ligase. It catalyzes the chemical reaction that joins the DNA sugar phosphate bonds. Okay. Restriction enzymes are sequence specific. They recognize and bind to specific DNA sequences. Okay, so this is just replaying it. Okay. Now we have DNA transformation, 2D animation. Stanley Cohen and Herbert Boyer inserted the recombinant DNA molecule they created into E. coli bacteria by means of a plasmid, thereby inducing the uptake and expression of a foreign DNA sequence known as transformation. Before Boyer and Cohen could isolate the recombinant plasmid they wanted, they needed a way to get their ligated plasmids into E. coli. Classic experiments by Oswald Avery and his group showed that pneumococcus bacteria are transformed to virulence when they take up DNA from virulent strains. However, natural transformation is a rare event, so they used a chemical method developed in 1970 by Mendel and Higa at the University of Hawaii. This involved mixing the bacteria and DNA in a suspension of cold calcium chloride at freezing temperature. Then, Boyer and Cohen rapidly raised and lowered the temperature to create a heat shock. This technique induces the bacteria to take in plasmid DNA. They spread the transformed bacteria onto a culture plate containing the antibiotics tetracycline and kanamycin. The only transformed bacteria containing both kinds of resistant genes could grow in the presence of both antibiotics. This technique induces the bacteria to take in plasmid DNA. They spread the transformed bacteria onto a culture plate containing the antibiotics tetracycline and kanamycin. Only the transformed bacteria containing both kinds of resistant genes could grow in the presence of both antibiotics. So they take the culture, put it on the plate that has both antibiotics, and then they start to grow. So these are all cloning. And here they are growing. This result was consistent with the bacteria being transformed with a recombined plasmid containing both the tetracycline and the kanamycin gene. So these genes were resistant. The result was consistent with the bacteria being transformed with recombined plasmid containing both the tetracycline and kanamycin gene. Okay. However, it is also possible that some bacteria had been doubly transformed by relegated versions of the original plasmids. Okay, so this is the original plasmid without the recombined DNA. And restriction analysis showed that some of the colonies had indeed been transformed by recombinant plasmid. Boyer and Cohen were able to tell which was which when they cut the plasmids and ran them out on an agarose gel. Roll over each band to see the difference. Alright, so we have recombined plasmid. We have this band, it shows, and this band. Okay, so in this section it covers the green and the red. And over here, green, and there, green. Okay. Down here is this red portion. Over here is this. This recombined plasmid doesn't have it. Then we have the blue and red. Over here is just all the blue and red on that small one. And over here as well. Up here is all red. And that's that. So here's just the PSC 101. PSC 102. This here is the recombinant plasmid. PSC 101. Boyer and Cohen had made the first recombinant plasmid. Several months later, they showed that these same methods could be used to recombine genes from eukaryotic and prokaryotic organisms. They inserted a frog gene into an E. coli plasmid. The resulting bacteria produced frog DNA. Again. Alright. Now let's go through the transformation process step by step. During rapid growth, the cell membrane of E. coli has hundreds of pores called adhesion zones. Okay. These pores are the adhesion zones. The cell membrane itself is made up of lipid molecules that have negatively charged phosphates. Okay, lipid molecules. They're all negatively charged. It's the cell membrane. made up of lipid molecules that have negatively charged. Okay, so there's the inside, here's the outside, and the adhesion zone. Even though the adhesion zones are physically large enough to admit plasmid DNA, the negatively charged phosphates on the DNA helix are repelled by those on the lipids. Okay. DNA plasmid. Okay. So it repels it. Negative and negative. So it's being repelled. Theoretically, calcium 2 Ca2 plus ions from added calcium chloride can complex with the negative charges, creating an electrostatically neutral situation. Okay. Also, lowering the temperature congeals the lipid membrane, stabilizing the negatively charged phosphates and making them easier to shield. Heat shock creates a temperature imbalance on either side of the bacterial membrane, which may set up a current. With the ionic shield in place, the DNA is then swept through the adhesion zone. As we saw, the DNA went from the outside to the inside. Heat shocks create temperature imbalance on either side, and that's what set up the current. So it's hotter out here and colder over here. Okay, so for transformation, I'd put technique used to insert recombinant DNA molecules into host cells. An example, plasmid DNA into E. coli bacteria. The technique induces host cells to take in and express the foreign recombined DNA sequence. The technique uses a combination of cold calcium chloride suspension and heat shock, raising and lowering of the temperature, to create a current that facilitates the travel of the recombined DNA through the host cell bacteria's adhesion zones on the cell membrane. Uses a combination of cold calcium chloride suspension and heat shock to create a current that facilitates the travel of the recombined DNA through the host bacteria's adhesion zones located on the cell membrane. Okay, that makes sense. Common DNA techniques: cloning, polymerase chain reaction, agarose gel electrophoresis, and DNA sequencing. A genetic structure in a cell, usually a strand of DNA that can replicate independently of chromosomes. An example, small circular strand, I'll put a small circular strand of DNA molecule found in the cytoplasm of a bacterium. Okay, so we continue here. We're going to look at common DNA techniques. Cloning, polymerase chain reaction, agarose gel, electrophoresis, and the DNA sequencing. First, the cloning of insulin. Okay. Synthetic human insulin was the first golden molecule of the biotech industry and the direct result of recombinant DNA technology. Currently millions of diabetics worldwide use synthetic insulin to regulate their blood sugar levels. Synthetic insulin is made in both bacteria and yeast. Okay. So how insulin is made using bacteria in a 2D animation. So in order to get bacteria to make usable insulin, a few tricks were used. First, DNA was made based on the protein sequence of the two insulin chains, A and B. So insulin chain A is this violet or purple, and this insulin B chain, which is blue. Okay. So let's see what the S's mean. Then an enzyme called DNA polymerase was used to make the second strand. These are the double-stranded DNA fragments that are inserted into plasmids. A chain, B chain. Okay. Each DNA fragment is inserted into the B-galactosidase gene on a plasmid. Here's our plasmid, the DNA fragment, A chain, B chain, or the DNA that was made from the A chain and the B chain. The plasmids also have tetracycline resistance gene. So the DNA fragment was inserted into the beta-galactosidase gene. So the beta-galactosidase gene was this more blue, dark blue, in contrast to this light blue here. So that fragment was inserted in between them. Okay. The plasmids are then transformed into bacteria. Tetracycline is added to kill off any untransformed bacteria. Okay, so then only the recombinant and transformed cells grow, bacteria grow. Okay. Plasmids are transformed, tetracycline added, kill off any other bacteria. So let's see. Bacteria normally secrete beta-galactosidase into their environment. The transformed bacteria are grown, then the beta-galactosidase and insulin fusion protein is harvested and purified. The beta-galactosidase part of the protein is cleaved off and discarded. Finally, the two protein chains are mixed together. Under the right conditions, the disulfide bonds, the S to S linkages, okay, that's what those S's are. I had a feeling that it was a sulfa. So the disulfide bonds, which means 2, 1, 2, so that's disulfide. These are disulfide bonds. Form and usable human insulin has been made from bacteria. Okay. Okay. Okay, so we're going to watch this movie here. So it's on DNA, synthesizing the DNA in order to synthesize human insulin using recombinant DNA technology. The human DNA sequence for insulin was needed. The amino acid sequence of human insulin was known. The Genentech group decided the human DNA sequence, or deduced the human DNA sequence of insulin based on its amino acid sequence. They then used the DNA nucleotides and synthesized the human DNA sequence. The sequence was then inserted into a plasmid and transformed into bacteria to produce insulin. By synthesizing the DNA sequence, the Genentech group assembled a human DNA sequence of insulin without ever having to use real human DNA. They thus bypassed some of the restrictions on human recombinant DNA work resulting from the Asilomar Conference. Asilomar Conference. So let's watch this. Whoops. Okay, so apparently there is no audio for it. So it just says, in order to synthesize human insulin DNA using the recombinant technology, the human DNA sequence was needed. Amino acid sequence of human insulin was known. Genentech group decided or deduced the human DNA sequence of insulin based on its amino acid sequence, and then used the DNA nucleotides and synthesized the human DNA sequence. This sequence was then inserted into the plasmid and transformed into the bacteria to produce insulin. By synthesizing the DNA sequence, the Genentech group assembled a human DNA sequence of insulin without ever having to use real human DNA. They thus bypassed some of the restrictions of human recombinant DNA work resulting from the Asilomar Conference, whatever that means. Okay. Vats used to grow bacteria. Still image. Okay. Vats used to grow bacteria. How was synthetic insulin first made? Although the idea was simple, in practice there were substantial problems. No one had the human DNA sequence for insulin, and there was a moratorium on the production of human recombinant DNA. Two different strategies were used to obtain the human insulin DNA sequence and transfer it into bacteria. In the end, one strategy and one company won out. Agarose gel electrophoresis, a technique for separating DNA fragments according to their size. Okay, we're going to look at agarose gel electrophoresis. Gel electrophoresis 2D animation. In the early days of DNA manipulation, DNA fragments were laboriously separated by gravity. In the 1970s, the powerful tool of DNA gel electrophoresis was developed. This process uses electricity to separate DNA fragments by size as they migrate through a gel matrix. Gel electrophoresis can be used to separate DNA fragments. Separate DNA fragments. Electrophoresis uses an electric current to separate different size molecules in a porous sponge-like matrix. Smaller molecules move more easily through the gel pores than larger molecules. Least path of resistance. While at Cold Spring Harbor Laboratory, Phil Sharp, Joe Sambrook, and Bill Sugden developed the DNA electrophoresis technique using an agarose gel made from highly purified seaweed. This could be used to separate DNA molecules ranging from several hundred nucleotides in length to over 10,000 nucleotides. Okay, here we have a plate of agarose gel, or a slide I should say, and those little white spots there are the wells at the top. Here we have a power supply. The gel is submersed in a tank filled with a salt solution that conducts electricity. Using a pipette, DNA samples are loaded into slots made in the agarose gel. The DNA samples are colorless, but researchers add a blue track dye. This makes it easier to load the samples and visually track the DNA migration through the cell. The phosphate groups in the DNA backbone carry negatively charged oxygens, giving a DNA molecule an overall negative charge. And in an electric current, the negatively charged DNA moves toward the positive pole of the electrophoresis chamber. From here, negative, to here, positive. The DNA molecules move through the gel by reptation, a reptile-like snaking action through the pores of the agarose matrix. Smaller DNA fragments migrate faster and further over a given period of time than do the larger fragments. This is how DNA fragments can be separated by size in an agarose gel. Okay, so we just wrapped up the electrophoresis. So in terms of the online assignment for the electrophoresis part, I have agarose gel electrophoresis, a technique for separating DNA fragments according to their size. The gel matrix is made from highly purified seaweed. The electrophoresis process uses an electric current to separate DNA fragments by size as they migrate through a gel matrix from the negative pole to the positive pole. The smaller molecules move faster and more easily through the gel pores than larger molecules. Visual inspection of the DNA reptation through DNA reptation movement through the pores of the gel matrix is made possible by using a blue dye. So the next part that we're going to talk about is polymerase chain reaction. Making many copies of DNA 2D animation. The polymerase chain reaction enables researchers to produce millions of copies of specific DNA sequence in approximately 2 hours. This automated process bypasses the need to use bacteria for amplifying DNA. And here, polymerase chain reaction. Amplification. Enter for a step-by-step guide to how DNA is copied in a PCR reaction. And then amplification graph. Enter to chart the number of DNA pieces or PCR products made for each cycle of the PCR reaction. Okay, so first we're going to look at polymerase chain reaction, PCR. Amplification. Polymerase chain reaction cycle 1. Let's see. The temperature is lowered to 50 to 65 degrees Celsius for 1 to several minutes. This allows the left and right primers to anneal or base pair to their complementary sequences. The primers are designed to bracket the DNA region to be amplified. Okay, so anneal primers, that means base pair to their complementary sequences. Okay, upstream primer, and this is a downstream primer. Okay. The temperature is raised to 94 to 96 degrees Celsius, denaturing the target DNA as in cycle 1. As before, the temperature cools to 50 to 65 degrees Celsius, allowing more primers to anneal. The temperature is again raised to 72 degrees Celsius. Taq polymerase binds to each priming site and synthesizes a new DNA strand. Third cycle, or cycle 3. We're going to denature the proteins, or the DNA. The arrow red in red denotes fragments of desired length. Okay, so here's a desired length, and here's a desired length. Okay. Anneal the primers. Now we're going to extend the primers. Okay, now the fourth cycle. Okay, so this strand is ideal. These 4 here and 4 here. Now we're going to go to the 5th cycle. Okay. Wow, 1, 2, 3, 4, 2, 3, 4, 5, 6, 7. Now we're going to view the graph. PCR amplification graph. Cycle, DNA copies, and target copies. So here we have the copies, and here we have the cycles. Okay, next cycle. So cycle 1, we have DNA copies are 2. Here we are, 2. Target copies are 0. Next cycle, DNA copies are 4. Okay, so it multiplied. Next cycle 3, we get 8. Target copies are 2. So in the blue, this dot. Next cycle, 4. Cycle 4, DNA copies are 16 because we were able to multiply it by 2. It doubled, right, from 8 to 16. But we went from 2 target copies to now having 8. Next cycle went from DNA copies 32, so we doubled them, 16 times 2, and we went from 8 to now 22 target copies. Okay, sorry about that interruption with the phone. So here we are, we have a PCR amplification graph. We're on the 5th cycle, we have 32 copies of DNA in the red, and target copies in blue, we have 22. We're going to go to next cycle, we have 64 and 52, 128, 114. Okay. As we see, here we are, 17 cycles, and now we have 131,000 copies and target copies. It's pretty dang close. And that's this purple line here, where they're relatively the same and there are only minimal difference between the two. But if we look, we'll just pay attention here. We're at cycle 21. We have nearly 2.1 million copies, and the DNA copies and the target copies, there's very minimal. There's only like a 42 copy difference between them, right? Now if I go to cycle 22, will it change? It should suspected to increase to nearly like 4 million plus, right? Yeah, there we go. So from every time it's doubling, and that's very valuable. 16,000 million here, watch, should be like 33 million. Okay, there we go, 33.5. Next, 66 or 67 million. Okay, bang. Okay, 120, 135 million. Okay, nearly, right? So every time it's, now it should be like 268, 269. 268 every time it's doubling. Final graph. Okay, there we are. 30 cycles, and there we are. 1,073,741,000. And still the difference is only like 100 something, because every time that we did it, it was very negligible between the copies of the DNA and the target. So we went through the amplification process, and we looked at the graph. I wonder why there's no audio for this. Okay, so PCR animation. PCR is a process where many copies of a specific piece of DNA can be made. This is known as amplification. Double-stranded DNA unwinds and separates when the temperature is increased. As the temperature is decreased, small starter sequences called primers, which are glowing, can attach or anneal to the DNA. These primer sequences are usually only 20 to 25 nucleotides long and are designed to match the start and end points of the DNA piece to be amplified. Once the primers have annealed, Taq polymerase blue copies of the DNA start from the primer. The temperature is increased and the strands separate. More primers anneal, the DNA is copied, and the cycle is repeated many times. In a typical PCR reaction, there are 30 cycles, which can potentially create 1 billion copies starting from one molecule of DNA. And we've seen that in the previous. Pretty impressive. when we watch the entire sequence and pay attention to the temperature increase and the decrease, and also to the number of DNA that are there, and it substantially grows. Okay, next slide talks about Sanger-Didioxy method for sequencing DNA. Okay, so we review the progress so far on the web assignment. We covered restriction enzymes, the tool itself, how it's used. We covered plasmids, the tool itself, how it's used. Ligase, the tool itself, how it's used. And then part 2 of the web assignment was techniques of the trade. Describe the purpose of each technique listed. And within the assignment was transformation, gene cloning, PCR, electrophoresis, and DNA sequencing. So we did cover all of them, except for DNA sequencing. We're about to start on that. But to review transformation, PCR, and electrophoresis, and I didn't end up typing in anything for gene cloning. I need to go back and do that. But the transformation was a technique used to insert recombinant DNA molecules into host cells. An example, recombined plasmid DNA into E. coli bacteria. The technique induces host cells to take in and express the foreign recombined DNA sequence. Transformation is characterized by using a combination of cold calcium chloride suspension and heat shock to create a current that facilitates the flowing travel of the recombined DNA through the host bacteria's adhesion zones located on the cell membrane. PCR, the polymerase chain reaction. The PCR process is used to create many copies of a specific piece of DNA. A single PCR cycle can enable researchers to produce millions of copies of a specific DNA sequence in approximately 2 hours. In a typical PCR reaction, there are 30 cycles, which can potentially create 1 billion copies starting from one molecule of DNA. And then in electrophoresis, the agarose gel electrophoresis, a technique for separating DNA fragments according to their size. The gel matrix is made from highly purified seaweed, and the electrophoresis process uses an electric current to separate DNA fragments by size as they migrate from the negative pole to the positive pole of the gel matrix. The smaller molecules move faster and more easily through the gel pores than larger molecules. Visual inspection of the DNA reptation movement through the pores of the gel matrix is made possible by using blue dye. Okay, and the next slides here we're going to cover DNA sequencing. Let's see, so Sanger-Didioxy method for sequencing DNA. This technique uses an enzyme called DNA polymerase to replicate DNA in a test tube. It also uses an unusual nucleotide. Watch the next animation to learn out what this nucleotide is and how it is useful. Okay, so we resume the Sanger-Didioxy method for sequencing DNA. Technique uses an enzyme called DNA polymerase, which replicates the DNA in a test tube and also uses an unusual nucleotide. Okay, early DNA sequencing, this 2D animation. First we'll read this here. The sequencing or two sequencing techniques were developed independently in the 1970s. The method developed by Fred Sanger uses chemically altered dideoxy bases to terminate newly synthesized DNA fragments at specific bases, either A, C, T, or G. These fragments are then size separated and the DNA sequence can be read. Fred Sanger's method of DNA sequencing was based on Arthur Kornberg's earlier work on DNA replication. Remember that a new DNA strand is synthesized using an existing strand as a template. Okay, to illustrate the technique, let's use polythymine DNA as an example. Okay. This polythymine DNA template goes from 5' to 3'. Okay. And that's what we see here. Let's see. Okay, so we have 3' end, that's where they're being added to. We have a thymine and an adenine, thymine, adenine, thymine, adenine. Okay. Let's watch it again. Boom, 5', 3'. Added there. Polythymine DNA template. Okay, polythymine, probably because there's many thymines here, right? So I just wanted to see one thing again. It looks like as they're bonding, I went back, that there's a an H2O molecule that is actually liberated, and you'll see that off to the right over here. Let's see. One is added, one gets, boom, look at that. What was that? ATP or something? H2O. H2O. And it looked like a phosphate. Something phosphate. Look at that. Long DATP. Okay. DATP. I'm just going to take a note of that. Right? That's what it said. Lowercase d, ATP. That one there, DATP. Boom. And it gives off two phosphates, and it looked like 6 oxygens. Okay, and that's where the phosphate backbone is helping form with the sugar. Okay. The 5' carbon of an incoming deoxynucleotide, DNTP, is joined to the 3' carbon at the end of the chain. Hydroxyl groups in each position form ester linkages with a central phosphate. In this way, the nucleotide chain elongates. So the 5' carbon of an incoming deoxynucleotide, DNTP, is joined to the 3' carbon at the end of the chain. 3' carbon, and here is the 5' carbon. The hydroxyl groups in each position form ester linkages with a central phosphate. Okay, here's the central phosphate and an ester linkage. Okay, I want to go back. In this way, the nucleotide chain elongates. Yeah. DATP. Boom. Another DATP. Boom. Okay, there's the phosphate and ester linkages. Alright, but as we go to the next slide, it says 5' of an incoming deoxynucleotide, DNTP. So I'm just taking note of those different nucleotides, DNTP. So we saw a DATP and a DNTP. I wonder what those are. Okay, we identified that. The key to Sanger's sequencing method is the peculiar chemistry of D-deoxynucleotides, DID-NTP. Okay. Like deoxynucleotide, a DID-NTP is incorporated into a chain by forming phosphodiester linkages at its 5' end. Okay. Did anything change with these photos? No. Okay. It looks very similar. However, the DID-NTP lacks a 3' hydroxyl group, OH, necessary to form the linkage with an incoming nucleotide, so the addition of a DID-NTP halts elongation. Okay. Okay, and here's another incoming one, but it's not attaching, or the chain is unable to elongate, and that's after one DID-NTP, dideoxynucleotides. Okay. To sequence DNA, 4 separate reactions are necessary. The first, one to provide sequence information about each of the nucleotides. Each reaction contains template DNA, a short primer, about 20 nucleotides, DNA polymerase, and the 4 DNTPs, one radioactively labeled. Oh, okay. This is why we saw one with an A on it. DATP, right? Let's see. Okay. So we have all those ingredients necessary to sequence the DNA in these test tubes. Okay, yeah. So to sequence DNA, 4 separate reactions are necessary. One to provide sequence information about each of the nucleotides. Okay, and that's why there's 4 separate. One for the A, one for the T, one for the C, and one for the G. Okay. Each reaction contains template DNA, short primer, DNA polymerase, and the 4 DNTPs, which one of them is radioactively labeled. Okay, so I figure, the radioactively labeled A, radioactively C, G, and T. That's my concept right now of this. One type of DID-NTP, A, T, C, or G, is added to each. Oh, okay. Okay. The D-deoxynucleotides. One type of D-deoxynucleotide is added to each. So maybe an A A C, a T, and a G. Okay, so it's not a separate DNTP. I want to know what that DATP is that we saw at the beginning. One type of D-deoxynucleotide is added to each. Let's focus on the adenine terminator reaction. So A, C, T, and G here. We'll focus on A. First, the DNA is denatured into single strands at near boiling point temperature. Okay. At high temperatures, the kinetic motion of the DNA molecule disrupts the weak hydrogen bonds that join the complementary DNA strands together. Okay. So temperature's rising. Okay. So they eventually separated. All right. And here we have the A. We're focusing on the D-deoxynucleotide labeled A. When the temperature is lowered, the primer binds to its complementary sequence in the template DNA, which was right here, I believe. Kornberg's earlier work showed that a primer is needed for DNA polymerase to start replication. Okay, decreased temperature, here we go, primer. Okay, the DNA polymerase makes no distinction between DNTPs or D-deoxynucleotides, DID-NTPs. Each time a DID-NTP is incorporated, in this case, DID-ATP, synthesis is terminated and a DNA strand of a discrete size is generated. Okay. Okay. We'll look at the process again. First, it's denatured. Temperature is increased. Strands separate. Lower the temp. Primer binds to the complementary sequence in the template DNA. Replication starts. In this sequencing example, okay, here we go. We have our answer, the DATP. In this sequence example, the D-deoxynucleotide for adenine in purple has terminated the reaction. The DATP happens to be the radioactive tracer, but this has no effect on elongation. And that's the yellow, the DATPs. Okay. So DATP is a DNTP. Understood. Because billions of DNA molecules are present, the elongation reaction can be terminated at any adenine position. Okay. This results in collections of DNA strands of different lengths. Okay. Okay. The same is true for the other three terminator reactions. Each reaction is then loaded into a separate lane of a polyacrylamide gel containing urea, which prevents the DNA strands from renaturing during electrophoresis. Ionized phosphates give the DNA molecule a negative charge, so DNA migrate toward the positive pole of an electric field. The movement of DNA molecules through the polyacrylamide matrix is size dependent, and this is similar to the agarose, but it's different in a way where now this this polyacrylamide matrix, this is actually a gel where the flow actually, let's see, so if this here was the contents of the mixture, and then it was actually put into the wells, it could be injected into the well, and this would be a dye, and it flows, it's a vertical flow. Okay, the agarose electrophoresis is a horizontal flow, but both are size dependent, and let's see, or it separates the DNA size, it's size separation specific, and the similarity, they flow from negative pole to positive pole. A blue dye that also migrates to the positive pole is added to the samples to track the progress of DNA in the sequencing gel. The dye runs just a little faster than the smaller fragments of DNA. Over the course of electrophoresis, shorter DNA molecules will move further down the gel than the larger ones. Millions of terminated molecules of the same size will migrate to the same place and band in the gel. After electrophoresis, the gel is sandwiched against X-ray film. The radioactive adenine in the synthesized DNA emit beta particles that expose the film, making a record of the positions of DNA bands in the gel. The sequencing gel is then read from bottom to top. The sequence of bands in the various terminator lanes gives the sequence of nucleotides in the template DNA. The sequence, okay, it's read from bottom to top. So this goes for an adenine, this for a cytosine, this is a thymine, and this would be a guanine. Okay. Oh yeah, and you look. So if we know that A and T, they bond together, or they're complementary. So look, the adenine here, and then the thymine. So we see like the T actually shows up in areas where the A doesn't. So in this adenine lane, it's blank here and blank over here, and we see thymine actually is here. So if we put them both together, it'd be like a solid line of full DNA. And the same would go with this cytosine and then this guanine. If we added this one with this, it would actually fill this gap, and the lane would be more complete. I don't know if that has any significance, but oh. So like this. Okay. TG T. Okay. There's no overlapping in the spaces. It's like for each band. Okay, I understand now. All of these put together would make one full line, would make a whole complete lane. So it's like in this space where we see this dark, it doesn't exist anywhere else left to right. In this one here, it doesn't exist left to right. This one, but if we put this one, this one, this one, this, then this, then this, then this, this, boom, 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 boom. That's how it's read, from bottom to top. Understood. Understood. A typical sequence reaction will yield 200 to 500 base pairs of readable sequence. Okay.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA. The first method of sequencing the genetic code was devised by Fred Sanger.
Okay. So it forms the basis of automated cycle sequencing reactions today. Scaling up the sequence in the 1980s, two key developments allowed researchers to believe that sequencing the entire genome could be possible. The first was a technique called polymerase chain reaction, or PCR, that enabled many copies of DNA sequence to be quickly and accurately produced. The second, an automated method of DNA sequencing built upon the chemistry of PCR and the sequencing process developed by Frederick Sanger in 1977.
To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
Okay. So we have DNTP is deoxynucleotide. And D-deoxynucleotide.
The first method of sequencing the genetic
Okay, so after typing out the purpose of, or completing the purpose of the DNA sequencing, what I'm going to submit on my assignment is this. The Sanger method of DNA sequencing consists of four separate essential reactions which sequence information about each of the nucleotides, A, C, T, and G. Together, the four reactions make it possible to carry out the main process of reading the sequence of an original strand of DNA. Or this, of a template sequence of a template strand of DNA. Each reaction is comprised of template DNA, a short primer, DNA polymerase, and the four DNTPs, or deoxynucleotides, one radioactively labeled in each reaction. Additionally, one specific type of D-deoxynucleotides is added to each. For example, one reaction receives DDATP, while another receives DDTTP, and the next one receives DDCTP, and the final one receives DDGTP. The DDNTPs halt elongation of the synthesized DNA fragments. Ultimately, because of the combination of ingredients with added heat for denaturation, this allows for the replication and termination of discrete sequences to take place. Ultimately, because of the combination of ingredients with added heat for denaturation, this allows for the replication and termination of discrete sequences to take place. The resulting collection of DNA strands are of different lengths. Each reaction is then loaded into separate lanes of a polyacrylamide gel containing urea, which then undergoes electrophoresis. After electrophoresis, the gel is sandwiched against X-ray film, and the radioactive deoxynucleotides from the synthesized DNA emit beta particles that expose the film, making a record of the positions of DNA bands in the gel. The resulting sequencing from various terminator lanes can be read from bottom to top. This sequence resembles the nucleotides in the template DNA. A typical sequencing reaction yields 200 to 500 base pairs of readable sequence. Okay.